Dear students, this is 12th Zoology video class, Unit 3, Reproductive Health, Higher Secondary, Second Year, Zoology, Part 7. Last class, we have seen what is cervical cancer, then what is infertility we have seen, then we have seen what are the other causes of infertility we have seen, then next topic is the one ART, assisted reproductive technology, that is we call as a ART. What do you mean by assisted reproductive technology? A collection of procedures which includes the handling of gametes and or embryos outside the body to achieve a pregnancy is known as assisted reproductive technology. What are the use of this one? What are the use of ART? This technology increases the chance of pregnancy in infertile couples. What do they say here? So here assisted reproductive technology are useful to increase the chance of pregnancy those who do not have child for so many years. So this ART includes intrauterine inseminations, intrauterine insemination, that is we call as a IUI, then IVF, in vitro fertilization, then ET, embryo transfer, then ZIFT, zygote intra Fallopian transfer, then GIFT, gamete intra-Fallopian transfer, then ICSI, intra-cytoplasmic sperm injection. So these are the methods are used in assisted reproductive technology. They'll ask you what are the technologies are used in ART. You have to write IVF, IUI, ET, ZIFT, GIFT, ICSI. So they expand also they will ask you what is the expanse of IVF. You have to learn this one word, important one word. And pre-implantation genetic diagnosis. Pre-implantation genetic diagnosis. Before they going to attach the embryo in the female reproductive part at the uterus, they'll find out the genetic disease, oocyte and sperm donations and surrogacy. These are the topic also we are going to see. We'll see one by one. First one, IUI. Before we enter into that one, I'll show you a few pictures regarding this ART technology where you can understand better. Just you can have a look at here. The picture what you are seeing is the one assisted reproductive technology. Here they take out sperm and egg from the body, outside the body, and just they make zygote or embryos. Once it's a zygote develop means this they transfer into female reproductive parts with help of different technology. That is we call as a ART. Here you can go through. These are the pictures. Assisted reproductive technologies. The next here IUI. IUI, this one. What do you mean by IUI? IUI means that's a intrauterine inseminations. Intrauterine inseminations. This is the image of IUI. You can go through this picture. What do they do in here? This procedure is to treat infertile men with low sperm count. Okay, the person one who cannot produce more number of sperm, such person they can extract the sperm from that male and just they introduce the female reproductive tract. That is we call as a IUI. So this is a procedure to treat infertile man with a low sperm count. The semen is collected either from husband or from a healthy donor and is introduced into a uterus through vagina by a catheter after stimulating ovaries to produce more ova. The sperm swim towards the fallopian tube. You can see this is a fallopian tube to fertilize the egg, resulting in normal pregnancy. This is one of the ART technology. Then second one, IVF. Second one is the one IVF. IVF here. What is IVF here? They say in vitro fertilizations. In vitro fertilization. Okay, here what they do in this techniques, the sperms and eggs allowed to unite outside the body. Outside the body in a laboratory, one or more fertilized eggs may be transferred into a woman uterus here. So this is the transferring here with help of this embryo transfer. So there you can find the picture, the bottom one, embryo transfer. Once the sperms and eggs are allowed to unite means join outside the body into laboratory. One or more fertilized eggs may be transferred into a woman's uterus. There may be many implant in the uterine lining and develop. Here after the embryo formed, that embryo may be just introduced in the female reproductive tract where they can go and just attach in the uterus. That's what they say. The excess embryos may be cryopreserved, which means the excess embryos, one or two embryos only just they introduce in this one, whereas others embryos where they can keep in a freezer one, frozen. That's this is a freezer, they're frozen for future use. In case if this embryo does not develop properly, they use the extra embryos. Initially, the IVF was used to treat women with a blocked, damaged, or absent fallopian tubes. Today, IVF is used to treat many causes of infertility. So, IVF, or we can also say test tube baby. We can also say test tube baby. IVF is also called as a test tube baby. Test tube baby. The basic steps is an IVF treatment cycle or ovarian stimulations. Ovarian stimulations. The ovary, they'll just start to release the egg. That is they call as ovarian stimulations. Egg retrieval, fertilizations, embryo culture, and embryo transfer. These are the techniques are used in IVF. The next retrieval is done by surgery under general anesthesia. Under general anesthesia. Now we are seeing in vitro fertilization. Here these are the explanations you can find in your book. You can go through in your book. Here the egg retrieval is done by a minor surgery under general anesthesia. So this IVF can be done by a minor surgery with giving anesthesia where they cannot feel a pain. For that purpose, anesthesia can be given using ultrasound guide after 34 to 37 hours of HCG. That's a human chorionic gonadotropin injections. The eggs are prepared and striped from surrounding cells. At the same time, the sperm preparation is done using a special media. After preparing the sperms, the eggs are brought together. 10,000 to 1 lakh motile sperms are needed for each egg. Then zygote is allowed to divide, form 8-celled blastomere, then transferred into a uterus for the successful pregnancy. The transfer an embryo with more than 8 blastomere stage into uterus is called as the embryo transfer technique. Embryo transfer techniques. They'll ask you one word. At what cell stage the blastomere can implant in the uterus? 8 blastomere cell stage. Important one word. The next topic is the one ZIFT. Zygote intra fallopian tube. Here, as in IVF, the zygote up to 8 blastomere stage is transferred into fallopian tube by laparoscopy. With the help of laparoscopy, when zygote develop into 8 blastomere, which means the cells just they undergo for the 8 cells. The zygotes they undergo for the divisions of 8 cells. At this stage, they transfer the 8 blastomere stage into fallopian tube by laparoscopy. The zygote continues its natural divisions and migrates towards the uterus where it is implanted one. The next topic is the one IUT, Intrauterine Transfer. The embryo with a more than 8 blastomeres is inserted into uterus to complete its further development. That is we call as a IUT. The next gift, gamete intrafallopian transfer. The transfer of an ovum collected from a donor into a fallopian tube. The transfer of ovum collected from donor of the fallopian tube. Ovum means what? That's a plural form of the egg we call as a ovum. In this stage, the eggs are collected from a ovaries and placed with the sperms in one of the fallopian tubes. The zygote travels to the uterus and gets implanted in the inner lining of the uterus. So this is because a gift. Then ICSI. I'll show you this picture where you can understand better. Here, this is a diagram of ZIFT procedure. ZIFT procedure. The 8 blastomere stage here. This is the blastomere stage. Can you find? Once the zygotes just develop into 8 cells, blastomere, and just they transfer to the fallopian tube by laparoscopy. Here they're transferring it. Can you see here? With the help of laparoscopy, just they transfer this 8 cell stage blastomere. The zygote continues natural divisions and migrates towards the uterus where it gets implanted. Get implanted one. The next one, that's IVF you have seen. Okay, and next, gamete intrafallopian tube. Just now I told you, we have seen the transfer of ovum collected from a donor into the fallopian tube. Transfer of ovum collected from a donor fallopian tube. In this egg, a collected from the ovaries are placed with the sperms in one of the fallopian tube. They're just placing the sperms into one of the fallopian tube where they can get natural fertilizations as zygotes. The next one is the one here. So this is we call as a ICSI. What do you mean by ICSI? Intra-cytoplasmic sperm injection. Intra-cytoplasmic sperm injections. You can find the parts which presents in this picture. This is the needle and the sperms. On the edge of needle, the sperms are there and eggs are there. Egg cells. Okay, here cytoplasm. They introduce these sperms into the cytoplasm. That's the methods of ICSI. In these methods, only one sperm is injected into the focal point of the egg to fertilize. The sperm is carefully injected into the cytoplasm of the egg. The egg, there will be cytoplasm will be there. In that cytoplasm, The sperms are carefully injected. The fertilization occurs in 75 to 85 percentage of egg injected with the sperms. The zygote is allowed to divide to form an 8-celled blastomere and the transfer into the uterus to develop the protective pregnancy. So this is the information about ICSI. That's the intracytoplasmic sperm injections. You can go through. Today we are seeing what is ART, the different technology are used in ART. We are completed. Just go through today itself. If you have any doubt, you can contact me through WhatsApp. With this, we'll wind up our class. Okay, bye. Take care.